and I hope you enjoy. Today we're going to talk about the future of genetics and advances in precision medicine with Rob genes. Just informing that Rob genes is a division, is using the technology of a Precision Group, which is headquartered in United States. We are the training division. So today I will start talking about the advantages of our systems and a little bit about genetics, just to start. So most of my presentations, my lectures, I like to say that we are 99.9% the same person. So in the world where we have billions of people, 99.9% from this billion of people, we are the same. Only 0.1% we are different. Exactly. So inside the 0.1% of differences, we have millions of genetic variations. So imagine that our body is very complex. And when I talk about millions of genetic variations, I'm talking about an average of 6 million genetic variations that make us different among ourselves. Okay, so this is fantastic. That's why it gives us different characteristics. But not only the color from our hair, the color from our eyes, our height, not only that. The most important differences that we have is how we survive, how we are prepared to survive in the world. And when I say survival or survive in this world, I say that we are different in the way we use the foods that we eat, in the way we metabolize the toxins, how we eliminate the toxins from the environment, how we deal with the ultraviolet rays from the sun. So this information is written inside our genes. Even the way we metabolize caffeine from coffee. For example, we have different types of people. Just for following the example of the caffeine, there are 3 types of people. People who metabolize very quickly, like me, for example. I can have caffeine and go sleep. I can have a coffee and go to sleep. 1/3 of people are like that. Another third of people, they are intermediate. Okay, they metabolize not so quick, not so slow, so the caffeine stay average inside the body. And the third type of people, the another 1/3 of people, they metabolize very slowly. So they are people who, if they take, for example, coffee 5 p.m., they cannot sleep because the caffeine stays in their body longer. And who gives this information? These 0.1% of differences that we have. Okay, so imagine 6 million different variations giving us a lot of differences, but not only bad things, good things also, right? Like intelligence, ability to deal with problems, stress resilience, so many things is inside our genes. So that's what I was explaining. We have 6 million genetic variations. Genetic variations could be, let's say in a summary, could be a mutation and a polymorphism, right? Polymorphism we know as SNP, SNP. What's the difference between mutation and polymorphism? Mutation is a genetic change, okay, something that is not expected to have in a population, but it's a change that happens in less than 1% of people. For example, we have 100 people, right? If there's one person with one change, this is considered a mutation because this happens in 1 or less than 1% of the population. Something like happens to in this 100 people sampling, we have 30 people with a specific characteristic, like for example, diabetes. This is not a mutation. This is considered a polymorphism because it happens in more than 1%. Okay, so understanding mutations are much rarer. It's not common to happen. Okay, so let's continue. Okay, inside our bodies, we have around 37 trillion cells. Exactly. At this moment, each one of us, we have 37 trillion of cells. Each cells performing different functions. We have cells in the liver, cells in the heart, cells in the brain, the neurons. They have different functions. And the total of 37 trillion cells. And the cells, they have a similar structure. These organelles inside the cells, for example, the nucleus contains DNA. And this DNA inside the cells are exactly the same, independently of the type of the cell. For example, the cell of a heart, the cell of a liver, and a neuron, they have a nucleus, and inside this nucleus, they have the chromosomes, and the chromosomes contain the DNA with exactly the same information, okay? So independently, if it's a hair, or if it's skin, or it's the lung, or heart, or brain, or pancreas, the information is the same, okay? So inside each cell, 23 pairs of chromosomes, making 46 chromosomes in humans, right? There are interesting things that rice, some types of rice, they have like 100 chromosomes, 110 chromosomes, make them a little bit more complex, right? But we are very complicated, humans, right? That's why genetics is so important, right? 46 chromosomes having a lot of information about our cells, okay? So it's inside our cells, we have 23 pairs of chromosomes. And what are the genes? People ask me, okay, DNA, genes, chromosomes, DNA, start to repeat, right? What's the genes? Genes are divisions in our chromosomes. Because if we open our chromosomes, we open it, we have, like exactly what I'm showing to you here, this image, we have divisions. And these divisions are considered the genes. So we have around 26,600 genes in a human body. 26,600. So before the Human Genome Project accomplished in the year 2003, we had not so precise information on how many chromosomes we had. Sorry, how many genes we had inside our chromosomes. But now, after 2003, we know we had more than 20,000, and today, to be more precise, we know we have 26,600 genes, and each one of them have a specific function, okay? Or they regulate the other genes, or they code for proteins. That means to make new proteins in the body. And when we have genes with problems in their function, and why happens the problems in the functions? Because you have a change in something which is expected, then something can happen. So let's go to the next slide. In the next slide, we're going to see exactly what happens when we have someone with a change, okay? So this is, we have, in our body, we mentioned we have chromosomes from the father and chromosomes from the mother, right? We have on the left side, the chromosome from the father, and the right one, the chromosome from the mother. That's us. That's why we have pairs, okay? Okay. If we see here on the left side, GATCAC in the father, and the mother, the same thing, GATCAC. This is what is expected. What is expected? Expected according to the population. So most of the people in the world, they will have this sequencing, okay? This sequence from the father and the mother. And this position here, this exactly, we choose this A here, this position. Let's imagine that this is the FTO gene. FTO is the gene for obesity, okay? I know very well. I have 4 mutations, actually 4 polymorphisms of the FTO gene. But imagine like this is the FTO gene, okay? And here is considered normal. What does it mean? That you don't have any change here. So you metabolize fats normally, your weight management is normal, okay? This is what is expected. So it's a normal, expected, expected. So here we have the right side up here, a normal phenotype is an individual which is weight is balanced, right? We have here A and A, can see here, A and A. It's considered what we call homozygous. What's the homozygous? Homozygous is when you have something equal, okay, from the father and the mother. But we can have homozygous of reference, which is homozygous, which is expected, right? We see here. And also you have the homozygous of risk, when you have changes in both sides. But this case here, it's what is expected. So what happens? This gene codes for protein. So there's no change in this gene. So the protein that will be necessary for our body will be normal protein. So that means we have a normal individual. Let's see a different scenario now. This we have an individual that instead of having an AA here, he has a C from the father and a C from the mother. You see here? C instead of AA. So that's been altered. When we have this alteration, we have what we call a polymorphism. It's a change, okay? It's a change. S&P, polymorphism is a change. So instead of AA, CC, it's also a homozygous, but this homozygous is a homozygous of risk. Instead of having a beautiful protein, we have a deficient protein, not nice protein. And then we have the phenotype of an overweight individual. You see that? And that's what happens in our body. So we can have also a mixture of both of them. We have a father, which has a normal position, normal allele, and a mother with a C in the position. Okay, so father is suspected, the mother is altered. Okay, could be the opposite, of course. AC we call heterozygous, minus and plus. In our graphics, heterozygous we show as yellow or orange. What's the difference between yellow and orange in our graphics? I'm going to show you later. The yellow is exactly this example we have here. We have someone, have a person that has one expected and one altered. It's called heterozygous. That means it's not expected because you have a change, right? But this change is not impacting in the individual. So we have here a normal weight individual. Okay, so this is what we call heterozygous of reference. In our maps, we show it as yellow. When we have someone which is a heterozygous of risk, that means in this case, this person would be overweight, then we have in orange. So because orange is closer, right, to the red. So it's a heterozygous of risk. In this case, it's normal. Okay, so it can happen. For example, not always that you have someone carrying one allele from one of the parents of risk that will develop that phenotype or disease or characteristic. Okay, so this is very important to understand. So you can have, according to our methodology of displaying results, you can have people who are heterozygous of risk, heterozygous of reference, homozygous of risk, homozygous of reference, and also protective. Okay, protective. I'm going to show you this later on. Okay, just to inform you, this is very interesting. It's about how many cells we make per second. We produce in our bodies more than 1 million cells per second. So let's count with me. 1 million, 2 million, 3 million, 4. Now I just produced like 5 million cells in exactly 5 seconds. Exactly that's what happens. But the cells are produced in a different quantity by organ that we have in our body. Just to give you an example here. In the brain and in the heart, we do not produce many cells. We produce more cells in the blood, okay? We produce a lot of cells also in the skin, right? Of course, in the intestines, but not in the brain and the heart. That's why we need to preserve a lot our brain, our heart, because our capacity of regeneration, of production of new cells takes time. Okay, of course, we produce new cells, but it takes time to produce new cells. For example, the heart, in the heart, we produce from 1 to 3 cells a day only, per second only. Okay, so it's not too much. Okay, but in terms of, for example, the skin, we produce from 30,000 to 40,000 cells per second of skin. That's why when we talk about skin, it takes from 25 to 30 days to have a completely new skin, right? So That's how we understand the production of the cells. It's very interesting to understand the regeneration of our cells. Well, let's start now, my favorite part. Methylation or methylation. Sometimes you're going to hear me say methylation or methylation, depends on my accent, of course. But this is how I learned about methylation years ago, okay, before becoming a geneticist. Methylation is, what does it mean, methylation? Methylation, if you got the word methyl, methyl, in chemistry, we know the methyl is CH3. It's CH3. This is a methyl group, carbon, hydrogen, okay? And the methylation is the act of giving methyl group to our DNA, okay? So what does it happen when we do the donation of methyl groups to our DNA? We turn specific genes off. For example, there are genes, different types. For example, I was explaining to you, you have good genes and bad genes. Okay, this is the first thing. You have, for example, the oncogenes, the genes that predispose you to develop cancer, and you have the tumor suppressor genes. The oncogenes, they should be turned off. Okay, so our body has the capacity of giving methyl group CH3 to keep those genes off, turn it off. Okay, other genes needs to be turned on, but it's not only about turning on and off the genes. Many functions in our body depends on the methylation process as well. Okay, so many genes, they depends on methyl groups to be performing their functions. We're going to see some things in the next slide, but this is the way I learned about methylation. And this map was, for me, was very complicated. Okay, so it was, wow, what is that? So, but I started years ago to understand better the metabolism in our body. And I found something incredible, that we have all our metabolic cycles connected. And I try to find out a way to display this to people, to show this to people, say, wow, we are connected. We are a machine. We are a big industry. Exactly. We are a big industry, big factory. We are all connected. So I came out with this. This is our metabolic pathway. I took years to develop that, years. And when you see that, you say, my God, like I did a lecture in Spain months ago, and the doctors in the lecture, they say, wow, how can I learn that? How can I understand that? It's very easy. Okay, I created this map. I used to say, I create a problem and I create a solution. And there's a solution to understand and how to interpret this metabolic pathway. So we're going to talk about that. You're going to see how interesting we are. Okay, this is a real case. It's a real person. Okay, and what we can see here. Look this. Looks how interesting it is. This is the metabolic, and this is what you can see inside the metabolic pathways, and very precise information. Just like a crystal ball, you know, like crystal ball, this we can understand people very well. So the first part, let's go to the top right, left, we have longevity area. On the top right, also longevity area. Okay, these are genes that have a very strong influence in the longevity process in the body. Okay, right here we have the dopamine area. Dopamine is a neurotransmitter related to movement, to interest, to movement, interest, pleasure, and this is a reward feeling. This is a dopamine. Here, serotonin. Serotonin is the happiness neurotransmitter. Also controls our gut motility, our sleeping. So this is we call the serotonin. Let's go back here. Yeah. So here we have the blood pressure area, where we have the control of our blood pressure. Thyroid area, here mental health, cortisol. This is super important. We're going to talk a lot about cortisol. It's very important. We have here the insulin and glucose. This would be the diabetes area, how we can understand. And what's interesting is the following. When we have a patient with diabetes, the doctors say, oh, probably you have insulin resistance, okay? But genetically, we can know exactly the reason behind your diabetes. If it's insulin resistant, or which gene, which variation is causing the diabetes in your body. So we'll be very precise in terms of diagnostics, and that means the right treatment for the patient. Alzheimer and dementia area, cardiovascular area, weight management area, inflammation area, cholesterol area, liver health, detox, mitochondria, which is our factory of energy. The CBS is the transsulfuration. This is a very important area. The GABA and glutamate, vitamin B12, folic acid, and methylation right here. So in this map, we have all these divisions. So when I see in the map reds, I see red points, that means it's alert. Let's take care. Let's see what we can do. Okay, so let's start understanding the metabolic pathway. So this is the latest way, the latest methodology of understanding one's body. Okay, so this is the best way of reading what's going wrong with the patient. But if you would ask me, okay, but how I'm gonna be able to analyze that? It's very quick. I'm gonna explain to you how to see it. Of course, I'm gonna do it more in details, but our system also generate a full report about everything which is written here. So this full report is good for doctors, for the patients, for lay people to understand what is written here, what the next steps, and what actions you need to take. Okay, so let's start. I'm going to change it, and I come back to this slide later on. So I'm going to show you this metabolic pathway. Okay, so I just generate a new version. I just tell you that came out a scientific publication last week, and I added a new characteristic, a new gene here in this function here as well. Okay, let's start. Every time we start analyzing, we start with this BH4. BH4. What's BH4? BH4 is a cofactor for several enzymes. Considering that our body is a kind of industry, it's like a necessary raw material for many functions that are performing in our body. So if your levels of BH4, that stands for tetrahydrobiopterin, is low, you are not able to do many metabolic processes. Okay, so first thing to check, how's your BH4 level? So let's check the genes that makes the BH4. So here I have green, 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 and green. So that means my production of BH4, it's okay. The BH4, when is donated as a cofactor for mainly 3 processes in our body, I'm going to talk about it, it becomes BH2. It's oxidized and becomes BH2. Okay, so this BH2, as you can see, is a cycle. This BH2 needs to be converted or regenerated back to BH4. And to do this regeneration, we need to have the function of 2 enzymes. The first one is the DHFR, and the second one, KDPR, that uses also in this pathway the PCBD1. We don't have any red here. See, green, 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 and green. So that means this person, when donates the BH4, goes back to BH2. Okay, so there is no problem in this cycle. So BH2 goes back to BH4, no problem. That means you have enough raw material, enough cofactor BH4. Now we are going to follow. Where goes this BH4 to? Okay, let's follow this in this line in blue here. Well, stop here. This area, as we saw in the PowerPoint, this is the dopamine pathway. Okay, we have here different colors. Okay, dopamine pathway. Why this is pink, not blue? Because it happens in the liver. Okay, this happens in the liver. So everything starts right here with the conversion of the amino acid. The amino acids are always in blue. Okay, when it's time you see this blue inside the square, amino acid, and the genes are inside orange. Okay, so it's here gene, here's a gene, here's a gene, gene, and gene. Okay, so for the phenylalanine, which is amino acid that we obtain naturally from the foods, for example, tofu, soy milk, chicken, beef, some kind of fishes, yogurt, eggs, peanuts. We need to convert in our body phenylalanine to tyrosine, another amino acid. Who makes this conversion is the gene called PAH. This gene converts phenylalanine to tyrosine. To make this job requires BH4. Okay, that's why I say it's a cofactor. Without BH4, you're not able to do this conversion. So what may happen if you have here a red? This person, if he has a red, would have a low levels of tyrosine. So that can influence where the tyrosine may influence the thyroid, right? As you can follow down here, the red line, you're going to see tyrosine plus iodine produces the T4, one of the thyroid hormones, okay? So sometimes we have people with problems in the thyroid, but the problem is not in the thyroid. The problem is right here in this conversion. So that's why I say metabolic pathways are, everything's connected. We can see problems. Sometimes they're very hard to diagnose, right? So we can see other places. Oh yeah, tyrosine, this case here is very well produced. There's no issue in this conversion, okay? Then we have tyrosine needs to be converted to L-DOPA. L-DOPA is the first precursor. And what's the precursor? What comes before the dopamine, okay? So tyrosine needs to be converted to L-DOPA. To make this conversion, we need an enzyme or gene called TH that makes this job. Again, TH also requires BH4. BH4 also is necessary for TH to make this conversion. So there is no problem in the BH4. We know that this person converts tyrosine to L-DOPA very well. Okay, so this conversion is great. Remember I said, what's the yellow? What's the yellow? Yellow is the heterozygous of reference. That means the father or the mother has a change in this SNP, in this polymorphism, but doesn't affect the phenotype, doesn't affect the individual. So you can consider this yellow as a green because it's not affecting. Here's different. Here we have on the right side is also is a heterozygous because it's an orange, but it's an orange representing a change in the phenotype. Okay, so this is closer to a red. Okay, okay. So this person, we understand, produces a very good quality of L-DOPA, and L-DOPA is converted through the gene DDC to dopamine. So what do we understand here? Okay, great. This person has a good level of dopamine in the production. Okay, but what happens with the dopamine? Dopamine is what we call a catecholamine. Together with the norepinephrine and epinephrine, the dopamine is following some processes. The first process that the dopamine, what happens with the dopamine is transported after production. It's like, imagine like in the industry, it's produced, needs to have a transportation, right? And every time we talk about a transportation or transport in our body, we need to understand there is a receptor for that. You cannot transport anything without a receptor, right? Imagine like a Federal Express, FedEx. It's going to transport something, but you have someone who is going to receive is a receiver, a receptor for this box, for example. And the same thing happens with the dopamine. So the dopamine will be going to specific receptors, okay? So we have here some receptors we call DRD1, DRD2, DRD3. Let's do something that I like to do, okay? First of all, so we have here DRD1, okay, the dopamine receptor 1. We have the DRD2. This is the DRD2. We have DRD3, and you have DRD4. Okay, you have here the main 4 receptors for dopamine. Okay, so each one of the receptors is related to some characteristics in this person. Well, that makes very interesting. Okay, the first one, let's go to the first one, DRD1. The DRD1 is related to attention deficit hyperactive disorder, impulsivity, addictions, aggression, antisocial behavior, bullying, impulsivities, and mood disorders. Okay, So it's very interesting that we find that. And what we see here, we have an orange and we have a red. That means this receptor doesn't work very well. So this person is very susceptible for these conditions here. Okay, how can we improve this person? The very interesting thing we're going to see, of course, in the big report, but here we can just have some ideas. Here we have, for example, the butyrate. The butyric acid, butyrate, which is formed in our body, in the intestines, through the fermentation of fibers. So that's why fiber is so important to eat, right? The butyrate, which is produced, helps to regulate the function of these receptor as well. So when we say, oh, we eat for our bacteria, that's correct, because the bacteria are going to help our body to produce the butyrate that will help to modulate this receptor, making this receptor to work better, okay? And then we'll have less side effects or less phenotype expression of these two variants you can see here, okay? Other one, DRD2, okay, DRD2. DRD2 is very important. Why? Because the DRD2 is related to anxiety and related to the mood disorder, okay? So you have someone, too much anxiety, too much mood disorder, change the mood a lot, probably you have issues with the receptor DRD2, okay? But there's something very, very interesting. There's a gene we call ANKK1, right here, see here, ANKK1. See here? This gene, when it's mutated, when you have here a red, you have a red here, that means you are very much predisposed to procrastination. And what is a procrastination? When you leave things to do later, I'm not going to do this now. Oh, I have to make a report now. Oh, do later. Okay, so this is the variation of procrastination, ANKK2, ANKK1. The NKK1 in this case here is a heterozygous of reference, so there's no influence. But when you find here red, you can ask the patient, you can ask the individual, do you procrastinate a lot? You're going to see interesting that matches very well. Okay. So procrastination is connected to NKK1. Okay, it's very, very interesting. Another variation, which is very interesting also, is the ADORA1. ADORA1 breaks dopamine in the DRD1 and 2. Okay, so what's the function of this gene, ADORA1? It's very interesting. We need to have a control of the entrances as well of the dopamine inside the receptor. It's not like, for example, in this case here, there's no mutation here, there's no change here in this patient. So that means it's free, so the dopamine can go inside as much as it wants? Of course not. In our body, it's important to have a balance of dopamine as well. Not too much, not too little. So which gene makes this balance is the ADORA1, okay? Why? It breaks down dopamine, allowing the dopamine to enter in the right quantity we need. But we have an issue here. This patient here, she or he has a variation in homozygous of risk. And what may it cause? Not enough breaking down of dopamine to enter the receptors. What are the side effects, I say like that, having this mutation? What's the consequence, the impact of having this? You may have Tourette syndrome, and also tics, okay, like people who blinks the eye too much or do repetitive movement. This is very common when you have ADORA1 variation, okay? So, of course, this all can be modulated, okay? So we have here L-theanine and curcumin together helps to modulate and helps this gene to make its function, its job, okay? Well, another variation that can happen is in the receptor number 3. Here's normal. When you have a red here or orange, is connected to ADHD, attention deficit hyperactive disorder, and one of the most important ones, DRD4. Okay, why? the DRD4 is strongly connected to attention deficit disorder and attention deficit hyperactivity disorder, both of them, okay? So having one red here, that means your chances of having ADHD is very high, okay? Even different levels, not, for example, not because you have this variation, you're going to have a strong ADHD, but you can have a mild one, and this can happen. So you can have 3 variations or just one like this patient, but it may happen. Okay? This is important to understand. So how can we, for example, according to the literature, and everything that I say is according to literature, we can address a patient like this. Okay, here's a list of some supplements, some food supplements. For example, the mucuna pruriens, which is a bean. It's amazing, it's a bean. Depends, of course, when you get in the market, depends on the percentage of levodopa you have inside. But usually when you have a high level, high quality and purity of mucuna pruriens, it does the job of the L-dopa. It's the same thing, okay? So it will increase your dopamine. That means why? When you have this variation, usually in genetics like that, when you have a variation and this gene or enzyme requires some cofactor or nutrient or vitamin to perform its function, means that you're going to need more. You are going to need more from this material, okay? In this case, DRD4 requires dopamine, but because of this variation, it's not going to use this dopamine properly. You're going to lose a lot of this dopamine, so you require more dopamine, okay? So it's recommended what? Mucuna pruriens. Mucuna pruriens, the first recommendation for this patient, so that you can give to the DRD4 more dopamine in the end of the day, okay? Another thing, bacteria, Lactobacillus rhamnosus. It's fantastic, okay, for that too. Citicoline, magnesium threonate. Magnesium L-threonate is a type of magnesium that penetrates the blood-brain barrier, very important. And rhodiola rosea, okay, rhodiola rosea, also very important for regulating the DRD4. So you can see other receptors, for example. The DRD2 needs different things as well. The DRD2, when you have mutations or variations here, I mean, Lactobacillus rhamnosus is the same one we mentioned here. Glycine, amino acid, look at the blue color here. Mucuna pruriens, butyrate or butyric acid. Remember I said it comes from fibers, from the fermentation fiber. Of course, you can find also in the pharmacies. Rhodiola rosea and phosphatidylserine and another amino acid, tyrosine as well. Okay, so combining all of this will help the receptor DRD2 to work better in case of variations. Okay, but not always you find needing for this, because in this case here, you don't need to do anything here. Okay, the problem is here. We have a red here that will create some issues, probably create some issues in the patient. For example, ADHD, impulsivity, and risk-taking. So people that sometimes takes too much risks without thinking twice, okay? Another thing that happens to the dopamine, we mentioned, let's go slowly, okay? We mentioned that dopamine goes to the receptors. We have the receptor 1, receptor 2, receptor 3, and receptor 4. We need to adjust the receptors, okay? We need to adjust making these receptors to receive the dopamine in order to make its function. But also the dopamine, which is produced in this cascade here that we saw starting from the phenylalanine, tyrosine, L-dopa, and dopamine, the dopamine cannot be so high in our body, okay? We have some enzymes or genes that makes the breakdown of this dopamine as well. So imagine the first step goes to the receptors, but also this dopamine needs to be broken down, because we are always eating, our body is always producing new amino acids that are going to be converted into dopamine. So we need to have a balance. So this gene, we can also call it enzyme. Enzyme is something that breaks, okay, breaks down. This is one of the most important genes in our lives, okay? So when I talk about COMT, C-O-M-T, and I say that it's one of the most important because it has a very strong impact in many different things in us. Okay, this individual, this girl or man, is very lucky because this person is what we call intermediate COMT. What is intermediate COMT? This is yellow, right? Intermediate. It's someone who degrades the dopamine in a very intermediate level. So that means it's average. So this is a good way of destroying, breaking the dopamine, okay? But 1/3 of people, 1/3, okay, 30% of people, they're gonna be yellow, okay, normal. 1/3 of people, they're gonna be red, which is fast. 1/3 of people are gonna be slow. I know my one, okay? And you should know your COMT if you are fast, red, yellow, intermediate, and if you are slow, which is the blue one, okay? That makes a lot of differences, and I will explain to you exactly what happens, okay? When you are fast, okay, when you are fast, red here, instead of having a blue, you're gonna have a red here. What happens to you? You can see the map here. You have a quick thinking. Let's compare with the blue one, okay? If you are fast, you have quick thinking. If you're blue, you are more planning person. It takes more time to take decisions. Stress resilience. If you are fast, you have very low stress resilience. You're extremely stressed, okay? Slow, you have a higher stress resilience. Pain sensitivity. If you are fast, you have higher pain sensitivity. What does it mean, pain sensitivity? You feel more pains. So if you are fast, you feel much more pains. Okay, so people with migraine, the first step we should analyze in one person, what is your COMT? It's very associated with migraine as well. Psychiatric disorder like schizophrenia and other psychiatric diseases, the chances is higher on fast COMT, fast degradator. Fears. People who are fast have more fears than people who are blue or low, okay? Anxiety as well, higher, okay? Very high. When you have a fast degradation of dopamine, your levels of anxiety are very high. So when you find someone say, oh, I have anxiety and it's very high, so you can research if the COMT degradation of the dopamine is fast, because it's really common. Pleasure experience reduces. So people who are fast, they feel less pleasure. Okay, they feel less motivation as well. Physical endurance, in the other hand, is greater. So people who are fast, they have more endurance, they have more resistance. So this is interesting as well when we are analyzing sport people that make sports, right? Addiction vulnerability, addiction for drugs, for smoking, for gambling, for many things. Okay, we have people who are fast, they have a higher addiction vulnerability. Extroversion, lower. Okay, so people who are more introverted, who are fast. Okay, and slow usually is the opposite. Okay, not in everything. Okay, but also if you have slow, your chances of addiction vulnerability is also high. Okay, you can see high and high, but because we're talking about the extremes, right? But let's talk about how can we modulate that. Okay, what can you do if you find somebody which is red here? In this case, I found a patient which is yellow, but we could have one which is red. Okay, but there are some natural supplements that, according to the literature, and not saying that, oh, it's maybe it's pseudoscience. No. This is proven. Okay, it's proven, and we know that there are lots of references saying that. Some supplements, for example, let's go here, in this area. Okay, for example, we have people who are fast COMT. They have lots of benefits with quercetin. Quercetin is a senolytic, okay? Quercetin. So the quercetin makes this fast degradation to stop. So imagine it's like a car moving very fast and destroying everything from the lift. That's a truck, a big truck destroying everything from the quercetin removes the foot from the brake, makes it go a little less, okay, going closer to the intermediate. And that's what we want. We don't want people to be fast degradator. EGCG, green tea, the green tea, another thing, okay? Glycine, amino acid, that's why it's in blue, fantastic to reduce the fast. Lactobacillus plantarum, which is another probiotic, very good. Rhodiola rosea and luteolin. So these here, 6 items, they help people who are come fast to stop degrading so much the dopamine. And what other things happens when we find somebody who is fast? Come fast is connected to inflammation. See here, inflammation. This is very, very serious. Come fast is connected to weight gain. So people who are come fast, they have more probability to gain weight. And people who are come fast, they have higher cortisol. You see that? Have higher cortisol. And when we talk about cortisol, we can go directly to the cortisol area. This is the area of the cortisol. Okay, this is the area when we talk about the cortisol. I'm just making it here. Okay, this is the cortisol area. We need to check if you have here reds. And unfortunately, this patient has a red right here. Right here, has a red here. This red means higher cortisol in response to stress. We all have stress every day, especially children. It's true. We have stress when we go out home, when we're late for meetings, when we have to wait for Uber. We are stressed every day, and in different levels of stress. We have stress every day. So people who have these variations here, they have more cortisol produced because of the stress, and that causes spikes or peaks of stress, of cortisol in the body. And this cortisol, what happens, is a big poison. Cortisol is a stress hormone, and it's a big poison in our body. Okay, of course, it's good because it helps us to have reactions when we are in danger. Okay, but in most of the cases today, in the 21st century, I say that cortisol is a big villain. It's an enemy. Okay, first of all, why? Remember we said here that this person converts phenylalanine to tyrosine, tyrosine to L-dopa. Very beautiful, right? Very beautiful. But this person is a candidate for high cortisol. So that means the cortisol will act right here. Look here. Cortisol is acting here. And the cortisol will inhibit the conversion of tyrosine to L-dopa. So if this person is having a stress season or a stress moment, he or she is not going to be able to convert the tyrosine to L-dopa because the cortisol will prevent that. So that means your levels of dopamine, despite you have a beautiful scenario here of genes, your levels of dopamine is going to be low. Okay? Having a low dopamine, all your receptors are going to fail, and your COMT is not going to stop. Your COMT fast, if it's fast, is going to continue degrading. That will create a lot of anxiety in your body. You're going to have mood disorder. You're going to have all the symptoms, memory failure, and so many other things. So the cortisol is a big problem. We need to modulate that. We need to work on this. When we have a red here, it's a signal that we need to do something, okay? So another very important thing to understand, let me explain to you. I know I'm talking to doctors and you know a lot about cortisol, but it's very important to explain that the cortisol is something that we humans, and when we talk humans, we talk about animals. I don't like to say we are animals, but biologically we are. We have the cortisol as a protective hormone. Why? Because when our ancestors, and we talk about also animals, they are in danger, they have stress, and the stress raises the cortisol, okay? What does it mean? You need to survive, you need to fight, and your body needs to survive what's going to happen. We don't know what's going to happen, but we need to survive. So what's the first thing our body does? Our body keeps fat when it understands we are in risk. So it stores fat. So that's the reason why when you have high cortisol, you gain weight, because your body closes the ability of processing this fat or metabolizing the fat, and starts to store this fat, and you gain weight. Okay, cortisol is connected to obesity. Okay, and why it happens? Because it's our protection, that our body understands you need to protect yourself and keep the fat. Because when you, for example, if you are fat, okay, you have you are overweight, your BMI is high, and there is a lack of food, okay, your body is going to burn this fat. You're going to convert this fat into energy. Okay, the triglycerides are going to be broken down. We're going to make energy from that. Okay, if you are skinny, you don't have, your chances to survive is very small, because you don't have anything to burn. You don't have the carbohydrates, you don't have the fat. The muscles are going to be, of course, you're going to be broken down the amino acids, but the fat storage is good for survival. Okay, not saying about the risk of having fat in your body, but animals are like that. So cortisol makes you keep fat, and then you gain weight, and also cardiovascular diseases will come. Okay, so this is important to understand. And so what we can do when you have these spikes of cortisol, and this person definitely will have 100%, he will have. Okay, it's proven by the literature. You need to give to this patient a combination of phosphatidylserine, ashwagandha, and magnesium taurate. These three combinations here are very powerful, especially the ashwagandha, which is proven to reduce the cortisol. Okay, but again, ashwagandha is very hard to find a good quality one. Why? Because the ashwagandha is processed, then you lose the main ingredients. Okay, so ashwagandha, if you want to work, you need to have the right origin of the ashwagandha. Otherwise, it's just a placebo. Magnesium taurate, very good also for the heart. Magnesium taurate, very good. And phosphatidylserine. Phosphatidylserine has the ability to enter the brain, okay, and also penetrate the blood-brain barrier, helping you to have more control, to calm down, and so on. Okay, so this is very important to understand. Okay, now we are going to continue talking about two other degradators. We spoke about the COMT as a degradator of dopamine, but there are two other degradators. One is the MAO-B. You need to check this in your patients, MAO-B. And the third one is MAO-A. Okay? The MAO-A degrades serotonin and also dopamine. MAO-B degrades dopamine. Okay? Here we have blue and blue. That means slow degradator. Okay? As I said, it's not good to have a slow as well. Okay? Not slow is not something very healthy. So we need to have the balance. So that's very good. When you prepare, you observe that, and you prepare the treatment for your patient interventions, it's very good to check our report that tells you exactly what to do, because the report, you're going to read this combination and give to you the right decision that you should take. Okay, so now we quickly go to see the other pathway, which is the pathway of serotonin. We spoke about the pathway of dopamine. Now we're going to say about the serotonin. Serotonin related to happiness, sleep, and digestion. Okay, So people say, oh, it's the happiness hormone. It's not a hormone, it's a neurotransmitter, but many people say it's a happiness hormone. Okay. Everything starts with tryptophan or tryptophan. Okay, tryptophan we get from food. Which foods are rich in tryptophan? Turkey, chicken, cheddar cheese, pumpkin, seeds, almonds, peanuts, salmon, eggs, and tofu. Okay, these are main foods rich in tryptophan. Tryptophan, when you get from food, and people used to say, oh, in the Christmas, people are so happy because they eat turkey. Turkey is rich in tryptophan. Tryptophan will become serotonin, which is the happiness neurotransmitter. Okay, it actually is not like that, okay? Because I'm going to explain to you what really happens, okay? Only 1% of the tryptophan that we eat, only 1%, is converted to 5-HTP. Okay, only 1%. The other percentages, they are lost, and other 90%, they follow the de novo pathway for the production of NAD+, okay, in terms of mitochondria. So this is very important to understand that tryptophan also produces energy, gives you NAD+. Okay, so many people, they make a confusion about your wellness that your NAD+ bring to you with the serotonin. Okay, but this is what is important to understand. Okay, well, this 1% of tryptophan, we have here 1%, this 1% needs the function, first of all, of BH4, again, BH4 cofactor. So in this patient here, BH4 is working. So great, we can make this conversion. But to make the conversion, we depend on these two different enzymes, TPH1 and TPH2. You see that? The TPH1 is more connected to the gastrointestinal motility. What does it mean? The movement of the stool inside the intestine. Okay? It's very important. TPH2 is more connected to the mood. Okay? More connected to the mood. So both of them, they will be converting tryptophan or tryptophan into 5-HTP. 5-HTP is the precursor of serotonin. But just to understand in terms of supplementation, if someone wants to take a supplement, for example, I want to buy L-tryptophan or tryptophan, but I don't know if I buy tryptophan or 5-HTP. Well, just have in mind that 5-HTP is 10 times stronger than tryptophan, okay? 10 times stronger than tryptophan. So taking tryptophan is good. Okay, it's good. It's very important. But if you want to have a quicker result and more effective result, 5-HTP, if you are adult, of course, okay, not talking about children, is better option for you. Okay, but understanding this map, and you have here the cortisol variation, we understand that this person is, if he's on stress, is not going to be able to convert even this 1% of tryptophan to 5-HTP because of the cortisol, okay? Because the cortisol also inhibits these two enzymes to convert the tryptophan, the 1%, okay? So if you are in stress, you are not going to be able to have 5-HTP. If you don't have 5-HTP, you don't have serotonin. If you don't have serotonin, you stop many other functions in your body. For example, you cannot go to the bathroom, you start having constipation, your sleeping quality is going to be affected a lot, and so on. So let's go understand one important thing. When you have vitamin B6 here, means that without vitamin B6, you cannot convert tryptophan 5-HTP, okay? Basically, I'm going to tell you something. Without vitamin B6, most of the metabolic processes that involves amino acids cannot be performed. You need vitamin B6 if you talk about amino acids, okay? So many people, they do even bodybuilding and say, oh, I'm not growing enough. Okay, how's your B6? Are you taking B6? Are you checking your B6 levels? Because without B6, your amino acids, they lose power, okay? And this is important to understand. Okay, let's consider that this person is converting well. 5-HTP will be converted by DDC. This gene converts 5-HTP and also L-DOPA, and you have the production of serotonin. The same thing happens with serotonin. Serotonin is produced, but go to some places. It needs to go to receptors. Okay, so we have here receptor of serotonin. Okay, here we have receptor. Is about reabsorption, and we have something interesting here to show. Second, okay, your serotonin is production, right? It's produced here. After that follows the receptors. Okay, if you have issue here, you have indication here of L-theanine that will be helping with this variation. But what is this variation here? ASD stands for Autism Spectrum Disorder. Autism is not a monogenic disease. I mean, you cannot detect autism with only one gene. It's impossible. There are many genes that predispose you to the autism, and some genes more than others. For example, SYNGAP1, CHEK3, and there are also some genes which functions may affect the susceptibility for ASD. Here we have a heterozygous of risk. Remember, orange, heterozygous of risk. Okay, so this is a candidate, but also we need to check other genes to know the chances of this person to develop autism. That autism is a spectrum, means that you can have a person that never realized has autism and people who have more phenotype of autism. Okay, so this can be observed. Great. Serotonin, imagine, is produced. Serotonin follows another pathway. After produced, serotonin follows this pathway. Okay, and in this pathway here, we see it finishes with the melatonin. So every melatonin in our body, you know what melatonin does? Melatonin helps us to fall into sleep. Okay, and this ability of converting serotonin to melatonin is reduced as we age. So that's why when we get older, we have more problems falling asleep continuously in the night. Okay, you can sleep thousands of times a day. You can just sit down and sleep. This is a different thing, but your ability of having a good level of melatonin reduces. Okay, especially when you have a TPH1 one. Okay, this variation, which is the same variation we see here, it affects the melatonin production. So that means people with this variation will have sleeping disorder. Okay, so how can you improve that? Okay, again, remember I said when you have a red means you need more from something. Okay, so in this case here, you're going to need help to produce melatonin. So the most indicated formulas to increase your melatonin is not taking melatonin. Okay, if you take melatonin the next day, you may feel dizzy. You need to clean it from your body. Okay, melatonin cannot stay too much in your body. So I do not think it's a wise decision unless if you traveling, and then it's okay. You can have a 1 milligram, 3 milligrams, but not more than that. Okay, but in a daily basis, there is a doctor already told me, oh, I've taken 10 milligrams a day to sleep. I don't think it's healthy, okay, first of all, because you need to clean it from your body. So the best way to increase your production of melatonin is taking magnesium glycinate and tart cherry extract. Why? Because both, okay, especially the magnesium glycinate, participate in the synthesis of melatonin, the production of melatonin. Okay, so if you have issues on this pathway, like this girl or man here has, you need to take magnesium glycinate. Okay, so magnesium glycinate will help you in the production of melatonin. That's why magnesium glycinate helps you to sleep better. Okay, people with issues sleeping, they take magnesium glycinate. And just remembering, magnesium, there's only one type of magnesium. Okay, only one type. Magnesium is magnesium, Mg. But there's a process in chemistry that we call chelation. When we mix magnesium with other amino acids. For example, glycine. I mix magnesium with glycine. I have magnesium glycinate. And glycine, we know, helps you to relax, helps you in many metabolic processes. So you have a mixture of magnesium with glycine. That means it enters the glycine receptor in our body, which is more abundant than the magnesium receptor, making you to relax and sleep. Okay? Okay. So I just showing to you a little about the metabolic pathways, what we can see. Okay. Well, following down here, let's go down. We spoke about the dopamine, we spoke about the serotonin, we spoke about the cortisol, right? Now we have another gene that is very important as well, is the BDNF. This gene is super important when we talk about cognition, memory, neurogenesis, which is neurogenesis, production of new neurons, and focus. Okay? So this patient is very lucky, has a green and has a blue, and the blue represents creativity, high creativity. Okay, so he's not in danger of having dementia because of the BDNF when he get older, or memory issues and focus. Okay, but what is very important to understand that the serotonin here, the serotonin levels, that's why it's good to check how's your serotonin levels. And people say, my memory is bad. Okay, maybe it's not because of the BDNF, maybe because of serotonin, maybe because of your cortisol, because the serotonin, when it's low, and can be low because also because of your cortisol, the serotonin modulates the BDNF. So the modulation of this gene, which is responsible for cognition, memory, production of new neurons, and focus, could be affected by the low serotonin. Okay, so it's important to check. That's why everything is connected. Okay, so the cognitive process, even learning processes, depends on this. Can you help the gene BDNF to work better? Yes. First, check your serotonin. If there's a low level serotonin, it's recommended for adults 5-HTP, the right level that your doctor will recommend, and also the other supplements that will help the BDNF, and it's proven by science, like lion's mane mushroom, acetyl L-carnitine is fantastic amino acid, L-carnitine, resveratrol, quercetin, and magnesium L-threonate. Magnesium threonate only in the morning, okay? If you take magnesium threonate in the night, you're not going to sleep. And if you sleep, you're going to have very vivid dreams. It's not good, okay? Because the magnesium threonate enters the blood-brain barrier, making you have a clear view of your day. You have more mental wellness. But if you take it in the night, you're going to have a very vivid dreams, and which is not good because you can really think it's a real thing happening, and you can have too much anxiety. Okay, this happens to many people. So it's very healthy, but take it in the morning. Magnesium threonate only in the morning. Okay, and this is the BDNF. Okay, it's very important to know what's your variation. If you have any variation, if you have red, your chances to develop cognitive issues is higher. But of course, you can modulate, you can help this gene variation with these supplements. Okay, okay. So now we are going to talk about the third participation of the BH4. I explained about the BH4 as a cofactor for this conversion, for the production of dopamine, for the production of serotonin, and also is used as a cofactor in the conversion of L-arginine amino acid to nitric oxide. So without BH4, you cannot convert L-arginine to nitric oxide. And why nitric oxide is so important? Nitric oxide is good for the heart. Nitric oxide also helps to regulate your blood pressure. Why? Because the nitric oxide opens the arteries, the caliber of arteries, allowing the blood to flow. So L-arginine is known for that. Why? Because L-arginine is converted to nitric oxide. L-arginine alone is not going to open your vessels. Who opens is the nitric oxide. But to make this to work, there is a gene called NOS3 or ENOS3. It's the same gene. This gene depends on BH4. It's a cofactor to make it work. So When you have the citrulline, which is converted to arginine, or you get directly the arginine, it's going to be converted to nitric oxide through the gene NOS3 and cannot have variations here. Here we found an orange. Remember I said orange? Orange is heterozygous or risk. That means is a change that comes from your father or from your mother that will impact the function of this gene. So that means this patient here, he or she will have a decline on the ability of the conversion of L-arginine to nitric oxide. So what the patient needs to do, okay, when he has that? First, check the BH4. Normal, great, good idea, good news. Other thing, we need to check the arginine level. And remember, when you have a variation, that means even if you have a good level of arginine, this NOS not going to use enough of the arginine. We need to increase the L-arginine a little bit, okay? Increase a little bit because it's not a red. In red, you need to give more L-arginine. Remember that amino acid requires B6, vitamin B6 as well, okay? Also, omega-3 is proven to help this function of the NOS3, okay, this enzyme. Vitamin C and vitamin E as well, okay? So what happens when you have a dysfunctional NOS3, means a gene that is not working very well? It's going to affect the memory of the person as well. See here, the memory is going to be affected because of a dysfunction in the NOS3. So this patient here has a heterozygous of risk. This heterozygous of risk will not give 100% of nitric oxide converted for L-arginine. This person needs more L-arginine, B6, omega, vitamin C, vitamin E, and also we need to check homocysteine and the levels of lead. You know, heavy metals, it's so important to check. Okay, why it's so important to check? If you have a high level of lead in your body, like I had before, high level of lead, you need to chelate it, you need to eliminate that, because lead also independently makes this NO3 to stop working. So independently, maybe you are great in everything, you can have green and green here, but you have lead in your body, you're not producing nitric oxide because your lead you have. Also, but if you have a high homocysteine is amino acid, we're going to talk about this. If your homocysteine is high, and this is one kind of exam that every doctor should ask to their patients, homocysteine. Why? Because if your homocysteine is high, also you reduce the nitric oxide conversion, the production, because the homocysteine stops this enzyme to work, okay? So you see, in case your lead is high, highly suggested methionine and glycine, okay, 2 amino acids to help to decrease your lead, okay, the lead in your blood, okay. That's as I did the treatment with this bowl, glycine and methionine, and my lead disappeared. Okay, so this is something that we can see in the map. Okay, just about the degradation of the serotonin. I just coming back a little bit more. I explained that the gene MAOA degrades dopamine and serotonin. Okay, it has a function. You can see degrades the dopamine and degrades the serotonin. Okay, so see here the patient is a green. It's a blue one. Blue means slow. It's a slow degradator. You say, it's good because it's going to have a higher levels of serotonin. It's not like that. The best scenario is always to have intermediate. Okay, what happens when you are slow MAOA, slow degradator of serotonin? You may have more mood changes. Okay, stress response, more stress. You're going to have more impulsivity. You're going to have more stress that can lead you to aggression. Look this, yes, aggression. Okay, doesn't mean you are like that. You are an aggressive person. No. You can become aggressive with stress. Good thing, psychiatric disorder, lower risk. Depression depends on environment only. So people who are fast degrader of serotonin, they have more chances for depression. Slow depends on the environment. Fears, People who are slow have more fears. Anxiety, more anxiety, and addiction vulnerability, more addiction vulnerability. So you see, having a slow is not good, okay? So it's very important to modulate this low as well in order to put it in an intermediate level, okay? To put it in intermediate level, okay? So this is all about balancing your degradation, okay? You need to balance the degradation. Not so fast, not slow. We need to go to intermediate. Okay, so there's a formula for each situation, combining many other aspects of the person. Again, returning about the, sorry, I just jumped from one session to the other. Now going back to the blood pressure. Okay, blood pressure, we started talking about the NOS3. Okay, but also we have this whole pathway. It started with the gene AGT. The AGT gene, and here we have heterozygous of risk. Remember, it comes from the father or the mother. The AGT is the gene that synthesizes, means produces angiotensinogen. Okay, angiotensinogen. And the angiotensinogen needs to be broken or cleaved by one enzyme which is produced inside the kidneys that we call the renin. Okay, so angiotensinogen is cleaved by the renin and then becomes angiotensin 1. Okay, angiotensin 1. See, after this process, angiotensin 1 is converted in our body into angiotensin 2. To make this conversion, we need one enzyme called AC2, ACE, okay, ACE. Okay. Here is where many beta blockers and many medicines for blood pressure acts. It cuts this. Okay, so that's where it acts. Why? Because when you convert too much angiotensin 1 to angiotensin 2, your chances of having high blood pressure is bigger. Okay, so here we have two variations, 4291, 4343. That means this person converts too much angiotensin 1 into angiotensin 2, too much of this. So that means a very high chance of high blood pressure, hypertension, okay? This is the first thing. Second thing, angiotensin 2, it stimulates, okay, what does happen here? It stimulates secretion of aldosterone, okay, the angiotensin 2, where it happens in the adrenal glands, suprarenal glands. And what makes this? Okay, regulates the sodium, potassium, and water, making or not your body to retain water and your blood pressure increase. Okay, this is a typical case of a hypertensive patient. Okay, but if you ask me, but look, this could be a child, and the child is normal. Okay, but this is what we call about the prevention. This can happen in this patient, okay, especially when you get older and your methylation capacity is reduced. Okay, so then you start losing the methylation capacity that will be controlling these conversions, and then your body gets dysregulated and your blood pressure goes so high, 17/10, and so on. Okay, so you have here very big risk factors for hypertension. Can you modulate that control? Of course you can, okay, even in a natural way. Understanding what's the problem, you can do the right control. Another gene that also predisposes you to hypertension, ADD1. It's a very famous one. ADD1 modulates your blood pressure as well. Okay, now let's go to my favorite part. Why I say favorite part, because it's the part about the methylation. Okay, so this is a complete chapter, but let's start right here. First of all, people make lots of confusion, and what's a folate, what's folic acid, what's metafolate? People think it's the same thing, right? And look, actually, the name is very similar, and there's a lot of confusion in the media what's the folate, folic acid? It's different thing, totally different thing. Now I'm going to teach you the differences, okay? But one thing in common between all of them is the vitamin B9. They are all vitamin B9. But vitamin B9, they have different shapes, different functions, okay? So let's start. Folate. Start with the folate. Folate is known as DHF. It's the same thing. Folate, DHF. Where do we find folate? Leafy greens, legumes, eggs, asparagus, citrus foods, beetroot. This we find folate in its raw format. Okay, so folate in a raw format is DHF. It needs to be reduced. So what's the reducement? We need to reduce DHF to become PHF. Okay? This process of reduction happens with the help of one enzyme called BHFR. It stands for dihydrofolate reductase. Okay, DHFR means reduces DHF. Okay, so this gene or enzyme has two functions. It's interesting. The first function, converting BH2, regenerating the BH2 to BH4. Okay, remember we talk a lot about that. Okay, and also has the function of converting DHF, which is the folate that we get from the food, to folic acid. Okay, now we start folic acid. Can we get folic acid from foods? Natural foods? No. But when you buy cereals like corn flakes and other unhealthy cereals, it's written enriched with minerals and vitamins, so on, you know that it's enriched with folic acid. Okay, they put folic acid inside. But in natural foods like fruits, vegetables, you don't find folic acid. Okay, folic acid is THF, which is converted from DHF. To make this conversion, you cannot have any red here. Okay, green means you do very good conversion. Otherwise, if you find any red, any orange, means that your levels of conversion to folic acid will be bad, not good. So instead of having 100% of the conversion, you're going to have like a 70%, 75%. Okay, great. After having the folic acid in your body, the folic acid starts what we call the folic acid pathway or the folate pathway. Okay, and this pathway like you see the wheel, right? So this beautiful wheel here depends on many steps, and each one of them are very important. So please pay attention. This is really, really necessary to understand. Okay, the first step, the THF, which is the same thing as folic acid, needs to be converted. See the square here. There's a green here, and we have another green here, right? The rectangles. Rectangle, three rectangles, right? This first rectangle, THF, needs to be converted to 510 MTHF. Okay? To make this conversion, THF to 510 MTHF, we need this enzyme. It's another gene. Okay? This gene needs to be operative. Okay? So when you have a red here, like we have in this patient, that means 30 to 40% less ability of converting folic acid to 510 MTHF. So that means if you have 100% of folic acid, I'm going to have around 60 to 70% of 510 MTHF. And what's the difference between that? Okay, the people say, oh, okay, no problem, eat more. Okay, you can eat more, but you're not going to convert. And you're not going to convert. This area here is so important because here is where we make DNA. Okay, this is what gives the raw material for the production of DNA. So if you cannot make a good DNA, good quality DNA, it's a big problem because diseases can happen. You're going to have lots of mutation. You cannot repair when you have a mistaken DNA. Our body has a repair system. You're not going to be able to repair your DNA properly. You see that? So when you have this variation, it's something to pay attention and do something, okay? Because this cannot be just ignored. For this process, your body will require vitamin B12. Okay, first of all, check your B12, okay? This is important. Second thing, I'm going to explain when you have this problem here, okay? There is one type of folic acid that we call folinic acid. And what's the folinic acid? Folinic acid is with one variation of the vitamin B9, okay, that doesn't depend on this gene that we're going to see here to be converted to L-methylfolate. So when you have this variation here, your body require folinic acid, okay, because this is going to help on this process, okay? You can give to you, of course, a little bit more folic acid, but if you give directly folinic acid, will help you with this variation that you have here. Combine it with choline and folinic acid, okay? To improve your DNA synthesis, glutamine and glycine. But here we have an exclamation point. DNA synthesis is important. For most of the people, yes. But people who have cancer, no. Then we need to be very careful. Why? When there's a patient with cancer, most of the chemotherapy processes work here. Okay, they work right here. Why? It's work to decrease this. Okay, stop the DHFR, the chemotherapy. There's a class of medicines we call the methotrexate. It acts right here, cutting the function of the HFR. Why? Because the idea of this chemotherapy is to stop the DNA synthesis in the cancer cells, right? So it stops converting the foods to folic acid. So then that means you do not have folic acid to be converted to 510 MTHF. So you cannot have a good production of DNA. That's the idea of the chemotherapy, to make the cancer cells weaker. Okay, this is the strategy. So when you have a patient with cancer, it's a totally different approach, it's a totally different aspect. So I'm talking about this conversion here for most of the patients who are not with cancer. Okay, cancer patients is a different approach. And also not only cancer, but lupus as well. Lupus, when you have a patient in a treatment with lupus, we need to be very careful because the medicines also be acting to cut this function here, okay? So then you can imagine, I put a parenthesis here, if you cut this function with the medicines, also you're gonna have less BH4, and then you have people who are with less dopamine, less serotonin, they're gonna have a higher blood pressure. So you see everything is connected, okay? So, but many things need to be observed. For example, I'll give an example, people who are in the treatment for lupus and they take a methotrexate that cuts this, they may feel much more tired. They can have a higher increase of having a stroke because their production of nitric oxide will be also reduced, and so on. Okay, so our report shows you, that's why it's important to, when you order the test with us, always tell us about how's your patient, everything you know about him, all the make a very good anamnesis, tell us what he's taking, if he's taking any medical drug, or if he has any disease or had before. Everything will be used by our 14 algorithms to calculate what to do, okay? So let's continue. Imagine now we have a patient who produces 510 MTHF, okay? Great. This gene here also affects the production of DNA, okay? So we have here a homozygous risk, and then it's suggested to give to this patient glycine and glutamine to improve the quality of DNA synthesis, because you have one problem here, and another problem here, okay? It will be important to increase this. But after having the 510 MTHF, our body needs to convert this one here into 5-MTHF, which is the same thing as methylfolate. Very important, okay? Methylfolate. So you see how we convert folate to folic acid to 510-MTHF, and 510-MTHF to 5-MTHF, which is the methylfolate. So this is now necessary, this very famous gene called MTHFR. This is very famous, and I tell you that has so much impact in our lives that you cannot imagine, okay? So in this patient here, we know already the quantity of 510-MTHF is not going to be more than 67%, okay, that we're arriving here. So we know it's not too much because we have this variation here. So when it arrives to be converted by MTHFR, it's going to use the 60% only. But we have a variation called 677. This gene has two main variations known by science, 677 and 1298. This is the name of the variation. This patient here has the 677 in red. So let's go here to the table. When I go here to the table, let's remove this. The table shows red, here red, and green, green. So I go here, I have 25%. What does it mean, this 25%? This is how much your body can convert of the 510-MTHF to methylfolate. So this is your production of methylfolate. And you need so much this methylfolate in your body, a lot. So imagine, if I have 100% of 510-MTHF, which is not this case, okay? You're only going to produce 25%. So you're going to have a very big lack, a big need of methylfolate. And you don't know it because you don't know your genes. What does it mean? That, look, this is what happens. The methylfolate has in the name methyl. You see the methyl? Remember in the beginning of this speech, I spoke about methyl. Methyl is what we call CH3. Okay, this is methyl. If I don't have methylfolate, I do not have methyl group in my body. And where I need the methyl group, where I need it. Let's follow this sign here. Now, follow this. Oh, right here, methylfolate. Now we are going to another pathway, which is the methylation pathway, or the methionine pathway. This methylfolate that comes from here, and I know that this patient here is only 25% is going to be converted into methylfolate, is extremely necessary in this process here, right here. And what does this process make? Okay, look this other wheel. We have one amino acid called methionine. We get it from food, okay? We get it from many foods. Methionine passes through different genes or enzymes until it's converted to homocysteine. Homocysteine in high levels is very toxic, it's very problematic in our body. It's amino acid, we produce it, great, it's important. Yes, it's important. But when you have deficiencies in this wheel, your homocysteine levels can be high. And when I say high, I say above 12, 15, 20. And what it causes? The homocysteine is a type of, when it's high, it's a kind of discontrol. The first thing we spoke, remember a few moments ago, it's going to stop the nitric oxide production. So that's going to make your blood pressure to raise, okay? First thing. And you don't know why your blood pressure is going high. And maybe because homocysteine is high. Second thing, the homocysteine makes injuries in the wall of your arteries. So it's like scars, make scars in the walls of the artery. When you make these scars in the walls of the artery, your autoimmune system is activated and starts to replace the scars with lipids. and making thicker. So your wall of the arteries, with the time, you're going to develop arteriosclerosis, and you can have a stroke. You can have lots of complications. And so this is a disease, a silent disease. You can see only through genetic test. Okay, you don't know what's going on. You don't know. Maybe you do a blood test. You know your homocysteine is high. You say, oh, doctor say, oh, take acid folic. You don't know if the folic acid is the right thing for you, because you, in this case of this patient here, you cannot take folic acid because the folic acid is not going to be converted to metafolate. So this folic acid, if I give to this patient, for example, folic acid, this folic acid, because cannot be converted to metafolate, will be converted as UMFA. What's the UMFA? And metabolized folic acid. That means it's a folic acid that cannot be metabolized. It cannot be converted to metafolate. But doesn't mean it's not going to participate in your body. Imagine how I like to explain that. Imagine like a bus, okay? A bus with 20 seats, like a school bus, 20 seats. Imagine that when you have a people with this variation here, you're going to say that you're going to have in the 20 seats, 15 seats will be occupied by folic acid that cannot do anything in your body, and 5 seats only with metafolate. So when it arrives at destination, the bus is busy. It's full 20 seats busy, but only 5 seats can do something. The other 15 are occupying space, and they're really disrupting functions in your body. So this patient here cannot have folic acid because it's going to be harming his body. And when you have, for example, a woman who is pregnant and the doctor, the gynecologist recommend folic acid, and he doesn't know that she is MTHFR 677 or 1298, or both of them, that also happens in heterozygosity, she is going to be creating a problem in her body because she's going to have unmetabolized folic acid. And there are many studies, I'm not going to enter this now, that says about the risks of having UMFA, unmetabolized folic acid, inside your body, especially for the baby, okay? But this is another chapter. So the metafolate, we need it right here. So why? Because the homocysteine that results from these conversions needs to be converted back or regenerated back to methionine. It's a cycle. You see a cycle. So homocysteine will become methionine, they'll become SAMe, become SA, homocysteine again, and go back. It's a wheel, okay? So when you have disruptions, and this disruption can be created because here I do not have enough metafolate. So that means without the metafolate, I cannot make this homocysteine to be converted to methionine back. Okay, so this is a big problem. If I don't have this, I cannot make this conversion, my homocysteine level is going up. It's going to be up. It's going to be working this here, but very low levels and making accumulation of homocysteine. So another problem that I see in this patient here, this patient has a variation called MTR. Okay, this variation needs much more metafolate and much more vitamin B12 to let pass the homocysteine. So when you don't have this functional MTR like we have here red, that means your homocysteine will even increase even more, because your body have issues to pass here because you don't have metafolate, you don't have enough MTR, and then demands more vitamin B12. So the body finds another pathway. This is we call the beta-in pathway. Okay, it's like an emergency exit that we call emergency exit. Because if we have a so high level of homocysteine, the impact in the heart is so bad. So the body has the other pathway. This is the beta-in pathway. And the beta-in pathway, what is so important to understand? What does it happen here? The homocysteine needs to reach the methionine. But to reach the methionine, the homocysteine needs to use beta-in. Needs to use this enzyme here. Okay, this is great. Okay, green and green, beautiful, no issue. Okay, but where the betaine comes from inside our body? Okay, we can, of course, you can have betaine from foods like from beetroots, from sweet potatoes, some shellfish, spinach, but it's not enough. Our body needs to convert betaine and needs to convert choline to betaine. And to make the choline conversion, we have here this gene, PHMT, that will use the choline and make the betaine. Okay, in this case, it's beautiful. There's no issue here. Also requires zinc to make this conversion. Great. But in this patient, look what's interesting here. Choline is a super important. It's like a vitamin. Uses the gene PEMT. The gene PMT picks up the choline, okay, and converts to phosphatidylcholine. Okay, so the gene PMT pick up the choline and puts in form of phosphatidylcholine. Phosphatidylcholine will act in the brain through the conversion to acetylcholine. Okay, so understand, this choline is going to the brain, preventing Alzheimer, preventing many bad things in the brain. Also goes to the liver, and then the phosphatidylcholine will allow the VLDL, the cholesterol VLDL, very low density lipoprotein, to be released from the liver. Without this, it stays inside the liver, creating fat liver disease. Okay, so in this patient is yellow, beautiful. Okay, no problem here. So we can go to next step. No, why? Because I found a red here. What's this red here? This is serious, okay, and this is something new, actually, that we just added in our system. This gene, CHKA, uses choline to produce phosphatidylcholine. Okay, when you have a mutation here or a polymorphism, as we can see here, it uses much more choline to produce phosphatidylcholine. So what does it mean? So this process, which was very beautiful and regulated, we see here, is disrupted by this. Because imagine, this variation requires much more choline. So the choline here that was supposed to be converted to betaine is not going to be enough. And also, this choline to be converted to phosphatidylcholine through the PMT is not enough. So this will be harming this process as well. Immediately, what we can understand from that, we're going to have a very bad methylation. We're not going to have even this way here to pass here, the homocysteine be converted will be reduced so much. So this patient necessarily and mandatorily requires choline. Okay, not only because we saw here it's choline, but here has this variation that requires choline, here also. Otherwise, what happens? Increases fat liver disease. You can even, this person could be skinny. Say no, he doesn't have any fat in the body. But even though because of this problem can increase the risk of non-alcoholic fat liver disease. India has 38% of people with fatty liver disease. Okay, so this explains a lot. Okay, so now the methionine is converted to SAMe. What's the SAMe? SAMe is like a vehicle, like a car, let's say a truck. This truck will pick up CH3, and where the CH3 comes from? CH3 comes from methylfolate. Okay, the CH3 from folate, methylfolate. And put this CH3 through the help of DNMT gene in the DNA, like here. Okay, so again, the methylfolate, we have in the name methyl. This methyl is CH3. In this process, methionine will be converted to SAMe that will pick up the CH3, and the CH3 will be put in the DNA, making This area of the DNA, the cytosine of the DNA in these positions, occupied by CH3, as you can see here. That means methylated. That means this gene is silenced. So when the RNA comes to copy the information of the sequencing here, the RNA is going to find, oh, this is busy, so I cannot understand that. Just like a book, we have a page with 1,000 characters in a book. So imagine you are deleting all the letters A, R, M from the book. You cannot understand what's written. So that's what happens when you methylate. You cancel this understanding of this sequence. So the RNA cannot understand. So this is very important. So the methylation process happens because of that. So going back a little bit, you see, this is so important. I said this is my favorite area. Everything starts here. So when you have problem like here, like you have 60% of this, then 25% of 60%, and then you don't have enough methylfolate, and then you have this issue here, require more methylfolate, more vitamin B12, and then you have HKA variation. It's a very big problem. So this patient can have a lot of problems. What happens when you have issues with methylation? You develop autoimmune diseases, allergies, you can develop different types of cancers because you cannot silence the oncogenes, hypertension, diabetes, women can have breast cancer, so many things. Can it be repaired? Of course, understanding the map, understanding the variations that we have here. And I saw other variations, like for example, this one here, TCN2. This gene is associated to the absorption of the vitamin B12. You can eat all the meat in the world. You can have supplementation of B12. You're not going to absorb that. So it's a problem because if you don't have B12, you're also not going to be helping this area to pass. You're not going to be able to recycle homocysteine back to methionine. Okay? And look at another thing. People with allergy, right? People with allergy. What's the relation of this methylation with allergy as well? Here, methionine, if it's healthy levels, if you have healthy levels of methionine, it stops the excess of histamine. So if you cannot have a good level of methionine because you have lots of problems here, you're going to have a higher level of histamine, and that means allergy reactions to pollen, to dust, to food, and so many other things. So you see how everything is connected. So, but don't worry, okay? I'm not telling you need to know by heart everything, because when we produce the report shows you all this information. So I know we don't have too much time left, so I'm going to jump a little bit quicker, and we can do more trainings like that. This is just a presentation, and I want to show more things to you as well. But another important thing that I would like to mention is about antioxidant capacity. And when we talk antioxidants, we talk about glutathione. Glutathione, it's so important. Why? The glutathione is the master antioxidant in our body. What's the oxidants? What's the reactive oxygen species? People talk a lot about that, right? That's true. I remember the first time I heard about that, and I became very interested in understanding what's that. And everything happens because of respirations that we have in our body, inside our body, okay? So let's go talk about a little bit about mitochondria. I promise I'm going to be quicker now, but this is going to continue, okay, another day. But mitochondria is our factory we have in each one of our cells, and responsible for making energy. Energy in our body we know as ATP, okay? You know that. Mitochondria, we have different number of mitochondria inside our body. In the heart cells, each cell of a heart, we have from 3,000 to 5,000 mitochondria. Imagine the quantity of energy the hearts demand during the lifetime of a person, right? So in the skin, you don't have too much mitochondria. In the skin, we have around 20 mitochondria per cell, but the heart, we have around 3,000 to 5,000 mitochondria. So that's why it's very important. And the mitochondria, when it makes energy, there is a respiration. We call it a respiration process. In this respiration process, is produced different types of oxygen, we call superoxide. Superoxide, okay, is O2 minus. This superoxide is converted to H2O2 by one enzyme called SOD2. So the SOD2 will pick up this superoxide, which is very toxic, okay? Superoxide, it's not because it's an oxygen, it's a type of oxygen. We call reactive oxygen species. So reactive means that reacts and can really cause damage to your cells, damage the mitochondria, okay? So not all types of oxygen are good. Even oxygen, we put oxygen with iron, you have what? You have rusting process, right? So the O2 minus, which is a superoxide, is very problematic in the body. So what do? We need to understand how is your ability in the mitochondria to remove, in other ways, to convert the superoxide O2 minus into H2O2 peroxide of hydrogen or oxygenated water, okay? This will later, if this gene is working, beautiful, because later your body will pick up this H2O2 and two enzymes will destroy it, will break it. The first one is the catalase, and the other one is GPX1, glutathione peroxidase number 1. So first of all, we have an issue in the mitochondria here, right? So this person is not able to convert the O2 minus, which is a superoxide, into H2O2. So it's reduced in conversion. So the damage that this person can have in the cells, in the mitochondria, lack of energy, because it's not going to be able to produce too much energy. He's going to feel fatigue and so on. It's high. So how can we treat this person here? First, we need to fix the mitochondria. Like what? PQQ, vitamin C, manganese is extremely important, alpha lipoic acid, coenzyme Q10, astaxanthin, L-carnosine. Okay, so salt helps. Okay, salt SOD helps because it comes from an extract from melons. But the most important here, we're talking about manganese, alpha lipoic acid, PQQ, and coenzyme Q10. This is going to really help this gene to convert the O2 reactive oxygen species into H2O2. Okay, eliminating that, just like a cleaner person, cleaning the mitochondria. But to cut this H2O2, which is also not good to have in our body, needs to be removed, okay? It's not so bad as the superoxide, but it's bad to have in our body, okay? You need to use the catalase enzyme, and the catalase enzyme, we see orange. This orange means is not working well. And if your homocysteine is high, you cannot even work, okay? So there's another gene called GPX1, glutathione peroxidase number 1, that will pick up the glutathione that we produce in our body, glutathione, and then uses glutathione to break this. And then it's okay. But unfortunately, this person also has a problem. He has a mutation, a variation, a polymorphism that doesn't allow these glutathione to be used to cut this H2O2 properly. So demanding more glutathione, he needs more glutathione, also needs L-carnosine, tartrate extract, selenomethionine, astaxanthin. This person is a big candidate for many problems because once you cannot cut properly the H2O2, chances of developing different types of cancer, including the prostate cancer, is high. Okay, so you need to start addressing preventative in this person. In terms of detoxification, we find here other things like GSTP1, GSTM1, so many other variations that it's about how you remove the carcinogens from the environment in your body. For example, plastic. Many people drink, I don't drink anymore, plastic bottles, water from plastic bottles, only glass bottles. Why? Because the plastic, you have particles of plastic. It is very toxic. And I need a very well GSTP1 working in order to remove it from my body. The same thing, the pesticides. Pesticides, right here. If this person has two variations here, you cannot remove the pesticides properly. It can be very damaging. The body can create cancer, okay? And xenobiotics, which are the medicines as well to remove. So everything comes from the glutathione. So the glutathione we're going to talk in the next course. Okay, we're going to continue from this area. I'm going back to my presentation. Let's go back to my presentation. Just to explain to you again where we stopped. Okay, we did what we did today. We spoke about dopamine, serotonin, BH4, cortisol, mental health, blood pressure, folic acid, the B12, methylation, and we start now with the detox and mitochondria. Okay, next time we're going to talk more about the detox, mitochondria. Then we continue to cholesterol, inflammation, diabetes, Alzheimer, and so on, many other things. Okay, so this is important to understand. This is a question I make for you. Which COMT are you? Very important to know if you have COMT fast, intermediate, or slow, right? In this case here, you can see a COMT fast person. And it's a totally different approach for this COMT fast. We need to take a lot of care of people who are COMT fast. And 1/3 of the population are the COMT fast, okay? This is another way that we can do. This is one of the reports. This report is a beautiful one from Precision about the skin aging report, where the system also recommends what to do to stop the aging process, anti-aging modulation. It's amazing. Even the recommendation of products that you can buy in stores and you can order that can really help your body, okay? And even the quantitative skin analysis results, this is only for skin. Imagine we have for every single disease, every single category of health. Even this person here, this woman, according to her DNA, chronological age is 34 years old. We have the biological calculation as well, but her skin apparent age is 32 years old, okay? And this is another pathway that also is very, very important, which is the fatty liver disease pathway, where we understand the risk of fatty liver disease, or if somebody is already sick, what needs to do to prevent becoming a fibrosis or a cirrhosis, which is not reversible. Here's the woman pathway about the peri and post-menopause transition. It's fantastic because you can understand for even the fertility issues. Some women, they cannot get pregnant, and or they want to reduce the symptoms of the menopause. It's very good. This is the cholesterol pathway. Is also fantastic and very, very important to understand. And how we analyze the result quickly and correctly and effectively. Also, we have a system called Imagine Care, where you put all the data from your patients inside, and then you can, even the blood test and everything. And our system makes the job for you, crosses all this information for you, your blood test, your image test, the genetic information from you, epigenetic information, and also the anamnesis, like the interview, and give you a beautiful report, what to do, next steps, recommendations, actions. So actually saves a lot of time for the doctors, and makes you have the right diagnosis for your patient in a 360 degrees analysis. So this is fantastic, okay? Integration of exams also we do, and the supporting material that we provide for the doctor. Very shortly, you're going to be releasing our book about the genes, the impacts, and so many other things. This is going to be for each one of you that subscribe and join us. This is what is actually an invitation that we want to do for the health professionals. If you are doctor, medical doctor, physicians, nutritionists, personal trainers, if you deal with health, if you work with people, like a pharmacist as well, we are looking for you. Okay, we need you. Why? Because when you use our test and our systems, you can really help people. You can really make the difference. Okay, it's not because I'm the head behind of Rob Genes and I'm the director of Procedure, but I tell you, we can make the difference. This is the objective. Every time we receive a testimony, and we have thousands of testimonies, positive testimonies from patients and family of patients, saying about how we improve through our tests, through our methodologies, their health, is a big pleasure for us. So we know what we're doing. Okay, we are different. We are not in the market to play or to be recreational like most of the genetic tests. we are doing a serious job. And this serious work represent years and years of research, a huge team. Only bioinformaticists, we have 96 in our team. And we have an amazing team of pharmacists as well, and doctors and nutritionists as well. And this is what we're inviting you to join us, start to use our system and methodology. Okay, so thank you so much. We'd love to hear from you. All the questions all the questions you have, we'll be more than happy to answer. And I'll be setting up a new seminar, a new continuation of this class for you too. So thank you so much. And for those who participate, there's a certificate for you that we're going to be providing as well. Okay. For those who are still here, I just want to show you one thing that is, people ask me, oh, but you have in your system 20 million scientific publications. It's true. Yeah, it's true. And what I tell you is something very interesting, that we have a tool. Look, this beautiful. You don't find it anywhere. That is our own tool that we know exactly what's your ancestry composition. We know exactly how much % you have from each origin. And why is this important? Why? Because unfortunately, most of the scientific publications, they come from US, or they come from China, or they come from UK. So can we use these publications for all over the world, for all over the population? Yes and no. And we know inside our system, what's your breakdown? We know exactly how much % you have, in this case, from Italian, how much you have from Iraq, how much you have from Saudi Arabia, how much you have from Spain. And then we apply the here from India, you see, and how much we can apply from this percentage in the publications that we have stored in our database. So we are extremely precise. Okay, so see you next time. Thank you so much for joining. So have a wonderful weekend ahead. Bye-bye.